Hello, I will be presenting this talk along with my postdoc, Dr. Aditya Narvekar, where we'll be sharing some updates with you on the N-glycanyzer workflow and how we've been using this platform for monitoring protein glycosylation and also amino acids to enable advanced and continuous manufacturing of glycosylated monoclonal antibodies. So let's get started. So the plan is to give a very brief update on my end, and then I'll have Dr. Narvekar take over and present the rest of the talk. So why do we care about glycans and glycosylation? Glycans are actually the building blocks of all living systems on our planet. It not only is important from the point of view that we see glycans all around us on the planet, it also covers all living cells. It also plays an extremely important role in how biologics interact with living cells, and hence glycosylation is an important critical quality attribute. So this is just showing you a little bit of a pictorial representation of some N-glycans that would be found on the surfaces of proteins and cells. So as I mentioned, glycosylation is a critical quality attribute of biologics like monoclonal antibodies. How we make these often are in bioreactors. There's a push towards making continuous perfusion processes as the industry standard, but often they're made in fed batch processes. Monoclonal antibodies have typically been made in mammalian cell lines, and often they have glycosylation in the FC region. These are just some representations of the common glycoforms that are found, or structures of the glycoforms that are found in humanized mammalian cell lines. Now, in addition to the glycosylation patterns that we are interested in, we are also interested in the cell culture conditions, often the media composition. And one of the key compositions of the media that impacts the glycosylation is the amino acid composition of the media. And often some of these kinds of spent media composition analysis is done on automated HPLC workflows, as shown in the bottom right over here, using some of these standard labeling chemistries with HPLC workflows from many of the industry vendors. Now, one of the challenges with glycosylation is that not only is it dependent on the process and the media, it also depends on the cell line, it depends on the cell culture conditions. This is just a representation of some of the important pathways of how glycosylation or N-glycosylation takes place in living cells. But the challenge is that if you are trying to control this, because glycosylation and the type of glycosylation that you have has an important on the efficacy of the drug molecule, we have to be able to control it, monitor it, and control it using suitable process analytical technology. So this is where my lab has been making advances in developing process analytical technology for monitoring glycosylation in near real time. And what I will now do is hand the baton over to Dr. Aditya Narvekar, who's going to present the rest of the talk.
Thanks, Professor Chundawat, for that brief introduction on glycans, the importance of analyzing CQAs like glycosylation and amino acids from the cell culture media, and further highlighting the importance of analyzing these CQAs during the process and therefore the need for development of process analytical technology tools. I will begin by talking about the process analytical technology, what exactly it is, and the types of process analytical technology tools that are being developed by various academic and industrial labs in the interest of the biopharmaceutical community. So process analytical technology is basically a tool which allows the measurement and control of a process based on the CQAs, that is, the critical quality attributes of the product, in real time or in near real time. Now, why is it important to analyze the different parameters of a drug product, like the glycosylation or the amino acid? It is basically important to have a good quality product. And are we not doing this already? Yes, of course. All the biopharmaceutical companies are definitely doing the analysis of this quality of the drug product. But in the conventional process, it is happening at the end of the production stage. In this case, we are doing while the process is going on, which means that we get to know about the quality while the process is going on and therefore take necessary decisions during the process, which further helps us to reduce the overall cost and the time of the product development and the manufacturing. The different types of CQAs can be at-line, on-line, and in-line for monitoring these critical quality attributes. Now, at-line analysis of critical quality attributes are when there is a physical separation between the pad tool and the process. Consider this arrow as the process, and there is clear physical separation as seen on the screen, which means that there is slight involvement of the technician during the process, where the sample handover from the process to the analytical tool is by means of a manual intervention. Moving on to online part. Now in online part, you have this difference of a single arrow in the downward direction. And that arrow is nothing but a denotion of automation, which means that the sample handover from the process to the process analytical technology tool is occurring in an automated way. Further moving on to inline part. In inline part, the process analytical technology tool is in line, as the name suggests, with the process that is happening, which means that the measurement is in contact with the process. Now, in case of inline analysis, basically these involves probes like Raman, IR, and near IR analysis for these kind of workflows. I'll be focusing on at-line and online analysis in the work that we are doing at our lab at Rutgers University. Moving on to the workflow that are being established in our lab. Now, what I'm going to talk about now is the proof of concept demonstration by the PhD student who previously worked in Professor Chundawat's lab. In this paper, they talk about the N-glycanyzer development. The basic workflow basically involves cell-free sampling, protein chromatography, denaturation of the protein, deglycosylation of the protein, followed by the labeling, enrichment, and analysis. The same thing can be seen in the form of a diagrammatic representation, where the sample is taken from the bioreactor. The impure mAb is then purified by use of a Pro-A column, then analyzed using a UV flow cell. Further, denaturation, deglycosylation labeling, followed by glycan enrichment and analysis. And the same workflows shown in the form of a pictorial representation can be seen by means of a flow injection device demonstration, which is actually the workflow that takes place in the lab on an instrument involving these valves and pumps, which ensure that different buffers, sample, elution buffers are going through different ports of the valves and mixing at the right time based on the command from the Python code in the software. Now, this workflow was basically developed using a well-known 2AB labeling chemistry for the glycan analysis. What are the pros of this chemistry is that the cost per sample is low, and it is one of the well-studied chemistry for labeling of glycans. The cons is that out of the whole process duration of 3 to 4 hours, the 2-hour duration is only the labeling, which means that in environments which are fast-paced, such as industries, it is difficult to use these kind of workflows. However, these workflows are very much suitable when you are developing processes, when you're standardizing such setups. It is quite economical to use these kind of chemistries to set up the workflow. And therefore, after the setup is done, moving on to expensive labeling chemistries like IPC is easier. So while this process sets us a benchmark for developing a flow injection analysis of glycans, it is not really favorable in industrial environments where time is a major constraint. In the next slide, I'm going to talk about a faster labeling chemistry using the Agilent's instant procainamide kit chemistry, where the same process as depicted in the previous workflow is there. You have the sample going into the Pro-A column, eluted out, neutralized with a buffer because the elution is at a lower pH, then denatured, deglycosylated, and labeled. Most important thing to remember here is that the labeling time. The labeling time you can see here is just 1 minute as opposed to 2 hours of labeling with 2AB. So that's the major advantage of this labeling chemistry, after which we send a sample for enrichment of glycans on an external guard column and then analysis using HPLC. However, considering that this is a flow injection analysis workflow and involvement of different solutions hooked on different valve positions on a flow injection device, you need to take into account a lot of things like dead volumes, length of the tubings, back pressure issues, or the column properties, the dimensions of the column. And if these things are not taken into account, replication of workflows is a challenge. And to account for the replication issue, we have made certain changes to an existing N-glycanyzer Python code in which we have included the easy input of tube properties like length, radius, etc. to ensure appropriate calculation of dead volumes, inclusion of column properties like the internal diameter, length, and column volume to ensure appropriate chromatographic profile, inclusion of detailed comments for each function to understand in a common coding language. Also, it's now easier to transition between labeling chemistries like 2AB and IPC. After making these changes to the Python code and updating it, this is the updated N-glycanyzer setup made at our lab. More or less, the setup is the same. We may have changed the position of the reagents on the valves. The major change that has been made is that taking into account the length of the tubings that connect between two different positions of the workflow. So if I'm connecting this central port of the valve to one of the ports on this pump, I will take it to account the length of this tubing into the Python code so that there is no issue in the whole labeling chemistry when the process is going on. It takes into account the dead volume calculations that are involved in the whole process. So that is one of the major changes that we have incorporated in the Python code. And therefore, specifying the length of all the tubes which are taking in reagents or which are connecting between 2 points is what is the major change here. Now, if I'm to give a reference to what I explained in type of PAT in the previous slide, so if I carry out the workflow in these 3 boxes here in the sky blue color, if I carry out these 3 processes, the purification, the Pro-A, and the labeling, only this process will be at-line process. And if I pick up the tube from here and place on the HPLC on the right, that will be the analysis part, where the analysis is taking place after I take the sample from the labeling pump and put it manually onto the HPLC. That is what at-line analysis is. You can here see external valve attached. I have made a diagrammatic representation of the same below, and that external valve is what ensures the entry of the sample from the outside to the inside of the HPLC. So I'm going to explain the at-line setup using this updated N-glycanyzer workflow and an online setup where even the sample handover is automatic to this external valve here. So moving on to the data using the 2AB labeling workflow. So this is at-line analysis. So the labeling is happening online. We are cleaning up the sample offline and then injecting onto the HPLC. So this is a representation of a triplicate analysis. These are the different glycans detected ranging from G0, G0F, MAN5, G1F, G1F prime, G2F. The top 4 glycans are detected. Along with those, the minor glycans like G0 and G0 and MAN5 are also being detected. So some of the changes that were made to the workflow, like staggered incubation instead of continuous 2-hour incubation, mixing between the steps, all those helped in having an appropriate setup of this whole workflow. The next step is obviously to try the entire workflow in conjunction with HPLC in an online manner. But before that, we will first see the at-line analysis setup with a faster labeling chemistry, that is IPC. Again, this is an at-line analysis, so the labeling is happening on the pump. There is an offline cleanup step, and then we inject onto HPLC. This is the sample analysis chromatogram with the at-line IPC setup. The glycans detected here are relatively more than 2AB. You can see the detection of G0F glucnac, G0, G0F, MAN5, G1, G1 prime, G1F, G1F prime, and G2F. And with that kind of detection, we further compare this data obtained with at-line analysis with that of the offline sample analysis. We get comparable relative abundances for all major glycans like G0, G0F, G1 prime, G1F prime, G2F. The relative abundances even for the minor glycans like G0F, glucnac, G0, MAN5, G1 were also similar. The variances, you can see it ranges from as low as 0 to as high as 0.8. And that 0.8 variance is for a less abundant glycan, which is G1 prime. G1 prime is one of those glycans which are detected at very low peak intensities, even with offline analysis. And therefore, detection of those peaks with an online setup using the flow injection device becomes a challenge. And therefore, work is being done in that direction to ensure that even the G1 prime glycan is taken care of. Although we do have detection, we just have to make sure that we are not losing out on its intensity. So after setting up this, the next part was after at-line to move the online. And to moving the workflow from at-line to online, as I described, the basic step is the automation of the sample handover from the PAT analysis to the HPLC. Now for sample handover, there has to be some communication between the devices. So the electronic communication between our flow injection analysis and the HPLC is by means of this software screenshot that I have presented on the screen and the device which is being represented here. This device basically acts as a connection between the flow injection analysis and the HPLC. Now to one of these ports on the relay device, we can hook up the wires from the HPLC and then we trigger the HPLC by means of the FIA device software and see if it is able to take the signal. So we did a lot of standardization in setting up the electronic configuration. We standardize which wire will act as a ground, which wire will act as a start request. We manually triggered the whole electronic communication between FIA device and the HPLC. And then after manual trigger, we tried it with a Python code to actually achieve that automation. And finally, all that was standardized. After standardizing the electronic communication, we obviously went on to try the complete online workflow where the sample handover happens to the HPLC, after which the sample is taken up by the external valve of the HPLC inside for analysis. Now with that, we don't really have very great results to present, but these are some of the crude results that we have in the initial trials. And as you can see that, as compared to the at-line IPC, in the online IPC, we are missing out on glycans, like the major glycans obviously are detected, but the minor glycans, G0, glucnac, and G1 prime are not detected at all. So that is the work in progress, and hopefully we'll be able to standardize this whole online analysis very soon with the external guard column sample cleanup. The other part of the talk, as you know, is about amino acid analysis. So I'll just touch base on it. Now, amino acid, as you know, that are analyzed from the cell culture media, and therefore we use the control cell culture media as a sample for analysis. Now, there are several things that has to be standardized while setting up this amino acid analysis workflow, like the sample transport solvent. The sample is going from an injection, the external valve. And when the sample is transported from the external valve to the auto sampler of the HPLC, it is important to have a transport solvent. So taking care of the right transport solvent, the sampling speeds, and other parameters, it's very important while developing online workflows. So using an external valve is challenging in some of these aspects. So after standardizing some of these parameters, we finally were able to have a chromatogram for CD-CHO media, which was comparable to that of the offline profile. And this is the identification of different amino acids that were detected on this chromatogram. And these are the retention times and the area under the peak for each of the amino acids. Although we don't have exact area under the peak as compared to the offline workflow, what we are interested in is to have the right peak profile and right relative abundances of the peak. And if those are achievable, the certain workflows, parameters which lead to sample dilution are acceptable. The only thing is that to have appropriate relative abundance of the peaks between the online and the offline workflows, which are achievable in this case. So with that, I will summarize the talk by stating the work done so far. So we have standardized the at-line 2AB analysis. Then we moved from 2AB to N-glycan analysis using IPC labeling chemistry. We standardized the protocol so that we have good glycan peak intensities. We fine-tuned the protocol further to even detect the less abundant or the minor glycans. Then the sample delivery with the complete online workflow was also tried using the external guard column setup, where the work is still in progress. And we also developed the amino acid analysis protocol using the external trigger. In future work, we aim to do both glycan and amino acid analysis in an online way by using two different external valves, standardize the whole method to have comparable peak profiles as standard offline method, and finally perform the PAT analysis after integration with the fed batch and the perfusion bioreactor. And with that, I conclude my presentation. Before I ask or open the floor for questions, I would like to acknowledge different lab members. This is a picture from a summer lunch at our PI's home. So I would like to begin with acknowledging my PI, Professor Shishir Chundawat, previous team members, Dr. Aaron Georgipal, Mr. Gokulnath Ganeshan, postbac, LSAM scholar, Ms. Nikita Brown, undergraduates, Jacob, Samir Parveen, Kendra Iris. Other lab members, Mohit, Antonio, Srividya, Ryan, and Austin. I would also like to thank our collaborators from Agilent, Mr. Wayne Haycock, Dr. Oscar Potter, Bob Geoffrey, Brian Whiteley. Special thanks to FIA Lab for all the help and the meetings regarding the Python workflows. And finally, our funding agencies for trusting us in developing this kind of workflows in academic lab, Nimble. So with that, thank you very much for your attention, and I'm happy to take questions, if any. Thank you.